Tēnā koutou, tēnā koutou, kura koutou katoa, and welcome friends and colleagues to the inaugural professorial lecture of Professor Ken Ryan from the School of Biological Sciences of the Wellington Faculty of Science. My name is Professor Grant Guilford, Vice-Chancellor of Te Herenga Waka, Victoria University of Wellington, and it's my honour to host you all here tonight. Professor Ryan has over 40 years' experience as a top-class research scientist. After completing his PhD at this university in 1980, Professor Ryan went on to work as a scientist at DSIR and Industrial Research Limited before returning to the university in 2002. During his career, Ken has undertaken research in a number of areas, from Antarctic climate change biology to freshwater cyanobacteria to kiwi fruit pollination. Over 30 years, he has made many significant contributions to the study of Antarctica and climate change, including work that helped lead to the award of the Nobel Prize to the Intergovernmental Panel on Climate Change in 2007. This Antarctic research has included as many as 16 trips to remote areas of Antarctica, the supervision of 9 PhD students and 5 master's students from both New Zealand and Australia, and $50 million in funding from the Antarctic Science Platform. Professor Ryan has contributed to the international biology community in many ways. He is a member of a number of local and international scientific research societies and conference committees. He regularly publishes in leading international journals and has contributed to several books. Ken is heavily involved in community engagement. His research has appeared in the media many times, including a recent feature on his team and their time in Antarctica last year on TVNZ. He is always ready to educate New Zealanders on his areas of expertise through visits to schools, as well as talks at Rotary clubs, fundraisers and at Forest and Bird. He is also an active member of the science design team for the Scott-based redevelopment program. Professor Ryan's teaching record is excellent, with many students praising the support and mentorship Professor Ryan has given them throughout the years. He currently serves as Program Manager and Postgraduate Coordinator for several areas in the School of Biological Sciences, and he was also involved in the Lab Planning Committee for our new Biological Sciences building Te Tokyo Rata. Tonight, Ken will discuss the impacts of environmental change on sea ice microbial communities in the Ross Sea, sharing 3 decades of research into these communities and bringing together both his research as well as the research of his postgraduate students and international colleagues. He will share how these important microbes, which act as a crucial food store for marine life, provide fascinating insights into future environmental change, acting as the proverbial canary in the coal mine due to the many challenging and unique stresses they face in their Antarctic environment. Ladies and gentlemen, please join me in welcoming the wonderful Professor Ken Ryan to the podium.
Well, thank you all for coming. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome everybody, friends, family, colleagues, special guests, our Vice-Chancellor, Professor Grant Guilford, our Pro-Vice-Chancellor, Professor Essan Mesbah, and Provost, Professor Wendy Lana. Thanks again, everybody, for coming. I'm very pleased to see such a crowd turning up for me. So I thought I'd begin. Let's just get this pointer showing a nice red pointer. So I thought I'd begin by acknowledging some 2 very important colleagues that I've worked with for many, many years. Firstly, Professor Andrew McMinn from the University of Tasmania. We've been working together in Antarctica for probably about 25 years and published lots and lots of papers, and I can't even remember how many we've published, so we're very proud to be part of a collaboration with him. And the second Andrew is Andrew Martin, who I hope is somewhere in the audience. I'm not sure. Oh, there he is. Yeah, hi, Andrew. And he did a PhD with me. He was my very first PhD student. And I'm very proud to acknowledge such an excellent scientist as developed from my mentorship. So I thought I'd tell you a little bit about myself. So I attended the Te Hapai program, and I managed to learn at the university how to finally pronounce the name of my little country town that I came from. It's called Matairi. We used to call it Matri, but Matairi is the proper pronunciation, so thanks to the Te Hapai program for that. And I imagine that none of you have ever heard of it. It's on the Forgotten Highway between Stratford and towards Taumarunui. So I did my PhD, as Professor Guilford said, at Victoria University, and I finished at about 1980, I should say, and joined the Physics and Engineering Laboratory out at DSIR, which has now become the Callaghan Institute. And when I was there, I was surrounded by physicists, engineers, chemists, and hardly any biologists. So I was a token biologist in lots of different programs, and we spent a lot of time just chasing money, different sources of money for different topics. So electron microscopy, X-ray analysis, wood preservatives, all sorts of different things, and stumbled across Antarctic algae and climate change, and quite a lot of other things that I can't really remember. Then came here in 2002 and have had excellent students working in lots of different areas, particularly freshwater cyanobacteria, and continued on with Antarctic algae and climate change. So, climate change. So you've all seen headlines like this. We're doomed in The Guardian. Tipping points. Sea level change. Carbon release. And just a couple of days ago, on Stuff, apparently Earth's going to be uninhabitable. In fact, there's a great article in New York Magazine, 2017, called The Uninhabitable Earth. If you want to read something scary about climate change, have a read of that one. And to give you a little bit of a taste, here's the opening line. It is, I promise, worse than you think. So that's been the focus of my research, is climate change, and been lucky enough to have a number of trips down south, some with the Australian program where I've managed to go on to various of the Australian bases, and with the New Zealand program in the Ross Sea. And one of the main places that I've visited is a place called Cape Evans, which is about 30 k's from Scott Base, I expect. And quite a nice place to visit, very beautiful spot. It's got Captain Scott's hut that he left for on his way south to the pole. And it's got a beautiful glacier, the Barne Glacier, and some very suitable sea ice that we've spent many years drilling many holes in. This is sea ice here, so we're standing on sea ice that's perhaps 1.5 to 2 metres thick. Now it looks pretty bleak, pretty barren, not much to see there, but if you go underneath and look back up, you'll see all of this stuff. Brown gunk, phytoplankton, bacteria, growing on the underside. It can get really thick. And if you look closely here, you can see these little bubbles. Actually, once you've got your eye in, you'll see bubbles all over the place. And that's oxygen, bubbles of oxygen. Phytoplankton are plant cells. They undergo photosynthesis, they make food, carbohydrate, and they produce oxygen. So it's a pretty important environment because it becomes what I like to think of as the grass of the sea. It's food for everything else in ice-covered regions of Antarctica and in the Arctic. Sea ice algae, really important ecosystem. So I talked about grass of the sea, There it is there. What's that beeping? Room will shut down in 50 seconds. Extend by 1 hour. There we go. That should be all right. So there's our grass, and these guys, krill, amphipods, mostly amphipods there, they're our sheep. And they're busy, upside down, grazing on this grass, munching away, and they become food for penguins, for fish, for whales. So I think it's a pretty important ecosystem. And some of the questions that I've been asking and what might happen under future climate change. And how does the sea ice microbial community respond to environmental stresses? So I've mentioned the word phytoplankton, mentioned the word photosynthesis a few times. Photosynthesis is a process that plants and phytoplankton use to take energy from the sun to make carbohydrate and produce oxygen. And in fact, if you total up all of the production of phytoplankton in the marine environment all around the world, phytoplankton produce about 50% of our oxygen. And they become the primary food source for pretty much everything else in the marine environment. Study phytoplankton, students out there. And they're generally green because they contain a pigment called chlorophyll. And that's where photosynthesis happens. You can even see it from space. So here's an image of the South Island here. You can see on the west coast, the east coast, you can see blooms of phytoplankton growing close to the coast. So how do we go about getting into that stuff? None of us are divers. So we generally drill down through. Sometimes we'll use a coring drill and drill right through from the top to get a complete ice core. Generally, we'll use what we call a jiffy drill. It's a post hole drill. This is Andrew, by the way, and this is Neville. Neville's our technician in SBS, so both of them have come south with me a couple of times. Andrew's been down nearly as many times as I have. And so we drill through the ice and go about 3/4 of the way through, put a coring drill in and core the last little bit. So this is our field site here. This is experiments we were doing in 2018. This is Mount Erebus. You've probably heard of Mount Erebus. Those of you may recall the Mount Erebus air disaster where a plane crashed about where the red dot is. They thought they were flying pretty much directly overhead. They were about 50 kilometres off course. Over here in the background is Scott's Hut. So it's a pretty historic place. So I've got a video to show you next. And some of this video was produced by some teachers who accompanied Andrew and I on our trip in 2018. And they did a virtual field trip for school children back here in New Zealand and did regular reports every day back to New Zealand. And they did lots of filming for us, so we've got some nice video. Now, normally we'd only use 2 people to do this drilling, but I can't remember who that third person is, but it was obviously somebody who wanted to be in the video. So here we go. This is what we do. We drill down with this drill, and then we use a coring drill and put it together, put it in that hole, and drill out a core sample from the last 50 centimetres or so, wrap it in a black blanket, put it in a black plastic bag and take it into the lab. There we cut the end off and do our work with this. This is Shelley, who is the teacher who came south with us, and she's making a much bigger hole using a hot water cutter, and that's hot water squirting out the end of that wand. Took us about 30 hours to drill through. We've got 6 pieces, there's one of them to take out. Once they were all out, then we put a tent over the top, and we've got a pretty scary hole in the ice. We had it covered most of the time, and yeah, it's a big hole because we wanted to put some instruments down through the ice, and I'll tell you about that a little bit later. So you can see some algae on the underside of the ice here. See pretty clear water down to the bottom. This is a camera going through the hole and looking back up underneath at the algae growing on the underside. This is video from a NIWA underwater robot with a pretty decent camera. It's looking up at one of the experiments we were doing in 2018. I'll tell you a little bit about that later. And these are a couple of holes. You can see the shadow of a tent here, and that hole often got visited by a Weddell seal, which is annoying. And this has got nothing to do with anything I'm working on. This is a Tina 4, but we think it's pretty beautiful, so I thought I'd show you that image. So here's one of those hunks of ice. And physical conditions in sea ice are really variable, which makes them a really good way of, a good working environment for us. It gives a range of different physical conditions, temperature, light levels and so forth. So near the surface, there are algae and bacteria that grow near the surface of sea ice. Not so many, but it's a pretty extreme environment. There's it's high light, it's quite salty water around the ice crystals, and it's really, really cold. Close to the ice-water interface, it's at the freezing point of seawater, which is about minus 1.8 degrees. So it's low temperature, but it's stable, it's low light, and we get a lot of biomass at the bottom. So a range of physical conditions all in one block of ice. So I want to tell you about some work that Andrew Martin did. So as I said, my very first master's and PhD student. And we'd decided we're going to do some work on the effect of light intensity on the growth rate of sea ice algae. And Andrew came up to me and he said, I'd like to do some work on the effect of light intensity on the growth rate of sea ice bacteria. And I looked at him and I started to think, I wonder whether I've made a mistake with this student. Because as you all know, bacteria are sponge grubblers, right? They decay stuff. They rot things away. They don't respond to light, Andrew. I may have rolled my eyes. So I decided, okay, fine. Well, it's time for a lesson for them. You know, you can't just ignore existing knowledge. You've got to build on it. That's what scientists do. So, okay, I told them, you're going to get a flat line. You're just going to get a graph, perfectly horizontal flat line, but let's do it. Good lesson for you. And so we did do that, and Andrew fed these bacteria from sea ice. He fed them a substrate. That's a two-part molecule. And if there are active cells present there, if they've got enzymes that are working, then the enzyme will cleave that molecule and release a dye that becomes bright in the microscope, and we can count metabolically active cells. So we did that. We spent 3 days, I think, doing this experiment. It was a long time. And we got this amazing graph. That was Andrew, by the way. So increasing light levels here and percentage of metabolically active cells, you can see a steady increase of bacterial cells responding to light, just as I said. And so I thought, here's a good opportunity to give them another lesson about science. Always question existing ideas. So that graph led to 4 PhDs and 2 master's theses. And those are the people there. So Andrew got a master's and PhD thesis from that work. The thing about this graph was that it set us on a path for new ideas. So perhaps the algae were responding to light, producing lots more sugars. Some of those leak out of the cells and feed the bacteria. That explains it. Or maybe cyanobacteria are there, phototrophic bacteria maybe, bacteria that can utilise light in a different way from normal photosynthesis. Normal photosynthesis? Oh, I should mention, by the way, we fairly quickly discovered that metabolic dye didn't actually work. It responded to light. We could have done that same experiment without any bacteria, and we would have got the same results. Ha ha. So, photosynthesis. Complicated reaction, in plants at least. I just put that up to scare you. Yeah, we're not going to do that. If you want to understand about photosynthesis, come along to one of my plant biology lectures. Some of those students in the audience will probably remember them. So we're going to talk about a light harvesting protein called proteorhodopsin. Proteorhodopsin, similar protein that we've got in our eyes. We call it rhodopsin in humans. Proteorhodopsin is a membrane-bound protein that exists in bacteria. So this is a bacterial cell here. And we've got this protein, proteorhodopsin, embedded in the membrane. Now, all it does is when light photons hit that protein, it changes its shape slightly. And as a result, a hydrogen ion or proton is able to pass from inside of the cell to the outside of the cell. That happens over and over and over and over again. And you end up with lots of hydrogen ions on the outside of the cell and not so many inside. We get a chemical gradient or electrical gradient. And those protons will want to get back into the cell. They can't get through the membrane. The membrane's impermeable to protons. But it will pass through another membrane-bound protein that's also an enzyme that catalyzes the production of ATP. ATP, for the biologists in the audience, will know that's energy molecule in cells. It drives just about everything in cells. So here we have a very simple system where light can be trapped in an energy molecule within the cell. We, at that time, when Andrew did his experiment, we actually knew about this compound, and we were hoping that we would find it in sea ice algae. Nobody had done, or sea ice bacteria, nobody had done that work anywhere in the world before. So we thought, particularly in an extreme environment like the top of the sea ice, those guys are likely to be growing there. So their presence and their significance in sea ice wasn't known at the time. So set a couple of students onto that sort of work. Eileen Koh, who's currently at University of Singapore, Rebecca Cowie, 2 PhD students, and they had a go at finding this stuff using different sorts of methods. And we managed to isolate a few clones, so there's one there. And we looked in sea ice and we found plenty of proteorhodopsin containing bacteria within sea ice. Most of them tended to be at the bottom, although we found them right throughout the ice. Now, recall that underneath here, there are sea ice algae. They've got chlorophyll in them, and chlorophyll reflects green. So we would expect these bacteria, if they've got proteorhodopsin in them, to respond to green light. So another student, David Burr, decided to do some experiments where he got some cultures of a known proteorhodopsin-bearing bacterium and put them in tubes and covered the tubes with different coloured plastics. And I think he drilled several hundred holes and put a tube in each one. After a couple of weeks, pulled the tubes back out, and we confidently expected that we'd get more growth in the green and maybe the blue coloured plastic tubes. And so that's indeed what we found. This is cell density here. The higher up that scale we go, the more cells have grown. If we grew them in the dark, not very many cells grew at all. If we grew them under red light, it was pretty much the same as being in the dark. They grew, but not so well. But we found that green and blue and ambient light all stimulated growth. So we were able to show for the first time that proteorhodopsin, not only was it present in sea ice bacteria, but that it was actually working under extreme conditions. And in 2019, we decided that working with cultures and tubes was not really relevant to the real environment, so we covered large areas of ice with different coloured plastics. And it was pretty much a preliminary experiment, getting ready and hoping for a Marsden Fund, which we have currently waiting on the outcome. If anybody in the audience on the panel, I'd just like to do a last-minute bit of encouragement to you. So we haven't got any results from that yet. We're still working on it. So that's what those panels look like from underneath the ice. These ones here are red, and you can see they're pretty much as dark as underneath the tent. So not much light gets through the red panels. There's the blue ones, and there's some green ones there. So leave the bacteria behind and have a think about the algae that I worked on. So how does sea ice algae respond to environmental change? We wanted to ask questions about their physiology. What happens at the cellular level? And then at a much larger scale, what is the biomass of this food? How much is actually available? How much food is available for the animals underneath the sea ice? So this is an experiment devised by Andrew. So despite all of those mishaps earlier on, Andrew and I are still working well together. And this is an experiment that he devised, and he called it the snow garden. And the idea was we'd lay out 5 by 5 metre plots and try and duplicate possible future climate scenarios. So here we've covered this 5 by 5 metre plot with about 30 centimetres of snow. And the idea there is that that's going to reduce the amount of light getting through to the algae underneath. The other thing that we didn't realise until we got down there is 5 by 5 metres by 1/3 of a metre equals 8 cubic metres of snow that we had to shovel around. Or I should say Andrew and Fraser did most of that. Naturally, as the team leader, I just supervised. So we have a low light plot here. We've got 3 of those. And a highlight plot, so we cleaned that right away so there was no snow on the surface. There's 3 of those. And a third plot somewhere in the middle. So it's about a 10 centimetre covering. And that gave us different light levels underneath the ice. We left them there for 10 days. And we thought they'll just get used to their new light conditions. And after 10 days, we drilled some core samples, pulled out the algae, had a look at what was underneath. And underneath the low light plots, we found that they were dominated by this particular diatom called Nitzschia stellata. And this is what it looks like here. That's this stuff, most of this brown stuff on here at the bottom of the ice. Nichia stellata in low light, but in high light we got Berkelea adeliensis, this one here. It's another diatom. It forms strands glued together with mucilage. So after our 10 days, we decided we'd shovel snow from the low light plot to the highlight plot. We did it there, that one went to there, this one went to there. Again, another lot of shoveling and a lot of work. We left these ones behind, come on up, as a control. So now this plot is going to have changed from low light to highlight. This plot is going to change from highlight to low light. So we did that one day. The next day we came back, drilled some holes, collected some samples, and did the same thing again 3 days later. And we found, to our surprise, that the previous low light that became a highlight plot, the species that was dominant now, after 3 days, was Berkelea. And in the highlight plot, Nichia stellata. So within 3 days, we could even see changes within 24 hours, but more pronounced after 3 days. So quite a dramatic change, and shows just how dynamic this system is, that if snow comes and falls on sea ice, then the species composition will change. As well as that, associated with that change in species, we decided to do some work with microelectrodes. So we had a number of different microelectrodes, one measuring oxygen. Now you all recall earlier on in the talk I was talking about oxygen being produced by phytoplankton. So that's going to measure oxygen production, phytoplankton growth. But we also had a number of probes that hadn't been used before in sea ice, hydrogen peroxide and nitric oxide. Both of those sensors measure indicator molecules for cellular stress. If those compounds increase in concentration, then the cells become stressed out. And this is work done by Fraser Kennedy from University of Tasmania, one of Andrew's students. So there's our ice core sample, and this water bath system here was made by Neville Higginson from our department. Here's our piece of ice here, and it's held down with some springs, holding it down in water. In this little bath, there's some filtered seawater. This pink colour is due to ethylene glycol, which we use to keep everything cold without freezing solid into ice. So let's have a look at what we got with that. If we didn't do anything, this is the plot we did nothing to, then the amount of oxygen produced didn't change. But if we have a look at this blue line here, moving from low light, relatively normal levels of oxygen production, we moved them to highlight, we got a huge increase in oxygen production. The highlight was stimulating photosynthesis, producing lots and lots of oxygen. And so we saw a large increase there, which dropped away fairly quickly. Now, high levels of oxygen can be very damaging for cells. It can induce the production of reactive oxygen species, and hydrogen peroxide is one of those. So that indicates perhaps the cells are going to be under stress, but let's have a look at what happened with hydrogen peroxide, and we get exactly the same result. Soon after, 24 hours after our transition, to the time when we shoveled that snow from one place to another, those that have moved suddenly into highlight are extremely stressed. Hydrogen peroxide, very high, 400 times higher than normal. And the cells manage to acclimate to these new conditions after 72 hours. We got similar results with nitric oxide. I'm not going to show you those. So our snow garden experiment showed rapid changes in species composition. That was quite a surprise. And the development of the appearance of that strand-forming bacteria, sea ice alga Berkelea, indicates, maybe changing light preferences have happened in that region over the last 15 years. 20 years ago, 25 years ago, I don't recall seeing that species ever. Significant physiological changes under light stress, that's obvious as well. The next step in the process would be to put these microsensors on an underwater robot, an ROV, see if we can get a larger scale view. And that's what we did in 2019, or should I say Andrew and Fraser, because I actually wasn't there, which is a bit disappointing because TV1 News, Caitlin Ruddock from TV1, visited the group last year, and some of you may have seen those clips on TV News just a couple of weeks ago. So I got in contact with Caitlin and managed to get hold of those clips, so I'm going to show you those. And I'll leave it over to Fraser to tell you what's happening.
I like the light just the right level, so some of them are going to be really stressed out, and we'll see that on these chemical traces.
Using microsensors, which for the first time are being taken underwater.
And because it's so game-changing, you can give it a go.
My gosh, no pressure.
The data is still being analysed, but already scientists are seeing species compete for energy under the different sheets.
There'll be some winners, there'll be some losers, but unless you have a dense community of algae at the ice-water interface, larval krill will struggle for a food resource.
Keeping a close eye on the smallest in the ecosystem. Caitlin Ruddock, One News.
So I've shown you these views underneath the ice. You can see these dense patches of sea ice algae. And we do cutting of cores. We take large number of core samples. And we don't know what it's like underneath the ice. We could be drilling through a piece here, or a piece over there, or somewhere here, or we might go through one of these holes. And every time we get a different estimate of the amount of biomass underneath the ice. So in order to get a decent understanding of the level of biomass in this particular location, we have to drill lots and lots of holes, and I can't even imagine how many holes that has been, probably thousands and thousands over the 30 years or so that would have been down there. So this is what we normally do. Oh, this is Simon Davey, by the way. Where's Simon? There somewhere. And Alice, this is your dad. That's him. Now, normally I would make jokes about how much taller I am than Simon, but this is an important occasion, so I won't do that. So normally we drill through the ice, we get our core sample, cut it up and so forth, but if we want to get onto a broader scale, how much is present over here, or over there, or in a different position? We can only sample on a small scale using this system. So I showed you this image before showing phytoplankton along the coast of New Zealand. Out on the Chatham Rise over here, you can see these blooms of algae, phytoplankton growing in open water. And that can be done with a satellite camera. Our eyes, digital cameras, your cell phone sees three colours, red, blue and green, and you interpolate with your brain the different colours. A hyperspectral camera is a different sort of camera that instead of just looking at three colours, looks at lots of them. 100, 200 different colours, different wavelengths of light. And you can do a process called hyperspectral imaging. And if you put that on a satellite and collect the light that reflects back off the surface of the ocean, then you can compare one region with another. If there's chlorophyll present, say here, for example, then some of those colours are going to be missing because the phytoplankton cells have absorbed them. So you can use that missing light to work out how much chlorophyll is present. You can do this with hyperspectral imaging, and the amount of chlorophyll that's present is directly related to how much phytoplankton is there. And if you do that, you get an image something like this, where red reflects large amounts of chlorophyll and lots of phytoplankton in the water. You can probably expect there's going to be some zooplankton eating it, and then there's probably going to be fish and lots of other organisms taking advantage of this highly productive zone. And you can see regions of high productivity and relatively low productivity in different parts of the country. So wouldn't it be great if we could use this satellite to look down at sea ice? But unfortunately, satellites can't see through sea ice. It's a bit of a shame. Fortunately, Andrew's got some friends over at University of Tasmania who have hyperspectral imaging systems, about $100,000. So we haven't got one, but Vanessa does. And she brought her hyperspectral camera imaging system down to Antarctica in 2018, 2019, and arranged a system where it's on a frame, a bit like Star Wars TIE Fighter, with some skis here, so we put some flotation device underneath the skis to hold everything buoyant and up underneath the ice. So the idea is that would go underneath the ice and slide along and look up using the hyperspectral camera, and we'll be able to get some images of chlorophyll distribution over a much larger scale. So this is a second clip from the news items on TV News a couple of weeks ago.
These algal masses help sustain the southern ecosystem, but they're hard to quantify.
So traditionally, survey and mapping of the Earth's surface is conducted from satellites, from aerial surveys, but one of the challenges of working with sea ice is that none of these traditional techniques are going to work.
Well, this is a classic way of accessing algae under the ice here in Antarctica. But one core is a fraction of the some 20 million square kilometres of sea ice around us. But this season, the scientists here are trialling new technologies to make this sampling area much larger.
It's the first time such a system has been used in polar waters, so the scientists are writing the handbook as they go.
So the engineering challenge was being able to get this hyperspectral payload, which creates a single line of pixels that scans underneath the ice to be able to be moved.
They built an underwater cable car. It's got a pulley system running a 30-metre cable along the ice. The camera is then fixed to a pair of skis and flipped over to face the algae.
This is one of the SWOTs that the system has scanned. These SWOTs are highly dense with information.
Ice core samples are still taken and are an important tool, but pixel by pixel, they're now building a map to a golden world below. Caitlin Ruddock, One News, Antarctica.
So this is the sort of result that we were aiming to get, so we're very pleased with this. This is a 20-metre transect underneath the ice, and it's about 0.7, 0.8 of a metre wide. And here's more or less a visual image showing different densities of sea ice algae on the underside of the ice. But this is a result of a hyperspectral imaging system where we're now able to say the chlorophyll concentration right here is 40 milligrams per square metre. Over here, about 15. In there, maybe close to 0. We've got a map of chlorophyll distribution on the underside of the sea ice. So we're hoping that this system can be used on a larger scale. There's lots of technical difficulties in getting it to work. One of the most important of which is there's not much light underneath there, and you need to collect a lot of light in order to create enough information in each pixel. So it's hard work, a lot of computing power, but we're hopeful that it might work for us in the future. So we think sea ice microbes are the canary in the coal mine, a window into the future. Thinking about warming climates, we might expect thinner ice, maybe some more rain, both of those likely to enhance light penetration, melting of the ice. In the short term, perhaps increased algal growth underneath the ice, increased productivity, more food. On the other hand, if there's more snow, it will decrease the amount of light that gets through the sea ice. Will decrease algal growth. We may change the species composition. It may not be as palatable to the organisms that want to eat it. If there's more open water and less sea ice, we'll get more phytoplankton growth in the open water, but the krill, juvenile krill, rely on sea ice algae as a barn for them over winter, and so we're likely to get less krill. So I thought I'd finish off. So I got a request from Simon Davies' daughter, Alice, that she wasn't coming unless there's going to be some pictures of penguins. So penguins, here you are, Alice. Penguins visited our camp in 2018. These are emperor penguins. And there is a rule when you're approaching wildlife in Antarctica, don't get closer than 10 metres. But if you sit still, camera at the ready, if they come up to you, that's fine. So they certainly did. So this is part of our camp here in 2018. So yeah, very lucky to be able to see those. This is Waddell seal and pup. One on the left is an Adelie penguin, and that's Andrew there, quite a bit younger Andrew. At this stage, Andrew was going through his maverick stage, so he fancied he was a pilot in Top Gun. So that's the reason for the glasses. And many of you know that I coach gymnastics in my spare time, and so it's not a bad handstand. Yeah? Not too bad, eh? Legs together, pointed toes inside the boots. It took a fair while to get that image, though. This is Captain Scott's hut. So our field camp is normally out over here. This is where we do our work. This is the hut, historic hut that Captain Scott used before he left on his fateful trip to the South Pole. That photo taken about midnight, so midnight sun there. So I draw the lecture to a close. Thanks particularly to the two Andrews and Vanessa, all of these students, lots of colleagues from New Zealand and overseas that have joined me down in Antarctica. And I think now is probably a good time to think about some drinks in the boat. Thank you very much.
Wonderful, wasn't it? Fantastic. Tēnā katoa. My name is Professor Essan Mesbah. I'm the Pro-Vice Chancellor of the Wellington Faculties of Science, Engineering, Architecture and Design Innovation. And it's my great pleasure to give the vote of thanks to Professor Ken Ryan for his wonderful lecture tonight. Shortly, we will be serving refreshments in the common room outside, but first I would like to highlight three elements from Professor Ryan's lecture this evening that I think exemplify the character and values of our leading scholars here at Herenga, Waka. But I have to go away from what I've written here. In my previous research, quite amazingly, I was chasing zooplankton, phytoplankton, and bacteria to kill them. Because when the ships go to different parts of the world, they carry these bacteria which belong to a different geographical position. And when they discharge them somewhere else, they become non-indigenous species and they cause a lot of damage. So having an insight into their life is fantastic because we finally failed. We couldn't kill all of them. So it was still a challenge for marine industry to be able to deal with ballast water management. However, the first, and as I have been here for just 202 days, I perhaps shouldn't still be surprised by this, but I very much am. The first is that my colleagues here in the Wellington Faculty of Science often seem to blur the line between scientist and intrepid explorer. When they say Antarctic research, they really mean it. They mean spending weeks and weeks in one of the harshest and most inhospitable environments on Earth. And everyone seems very relaxed and casual about this. And I think we very much noticed that tonight. They appear to be as comfortable drilling holes in the Antarctic ice sheet as they are in the lab. This reminds me of the movie Ice Age. I'm just looking at the little girl up there, that how many of these ice holes are created, made a lot of changes around the world, and we noticed it in that cartoon. This is something that continues to impress me, and I have huge respect for people like Professor Ryan for their commitment and grit when it comes to complex, large-scale field research. The second is the emphasis on community, both outside and within the institution, and in particular having such a welcoming and inclusive attitude towards students. I think it's very clear that Professor Ryan is an incredibly dedicated teacher and supervisor. For him, it doesn't seem to just be about imparting knowledge onto students. Rather, he seems to give them the opportunity to be part of groundbreaking research and to contribute and be part of the team. And I think that Andrew is a clear example of that. I imagine that being a student of Professor Ryan has been a life-changing experience for many young people. And thirdly, I am impressed by the breadth and application of Professor Ryan's work, and tonight's lecture is a perfect example of that. In covering 30 years of research, he has gone from the microscopic to the global. He has taken us from the bleakness of the Southern Ocean to the rich color and complexity of microbial life under the ice sheet, and used this to offer a window into the future. This is an imaginative and impactful approach to science and to research, and it is exactly what we need as we navigate the rocky waters of the global climate emergency we now face. So, Professor Ryan, thank you very much for your fantastic lecture this evening, and my warmest congratulations on your well-deserved promotion to professor. Thank you.